Thank you so much, and thank you, the organizing committee, for selecting me for this talk. So I want to talk about cancer. For many years, it's been called only the disease. Unfortunately, many of us know someone or known someone who had the disease. It was considered for so many years that it was fatal and has no cure. And it comes from the Greek word karkinos, a crab. Hippocrates termed it so because the veins stretching out from the surface tumors resembled crab legs for him. Unfortunately, this disease took over as the #1 death reason in Israel. For more than 70 years, we had only 3 treatment modalities: surgery, chemotherapy, and radiotherapy, similar to all. And chemotherapy and radiotherapy kills proliferating cells, cells that divide and grow, but like the cancer cells. But it also kills normal proliferating cells, cells like the intestine lining and hair follicle, leading to harsh side effects like vomiting and hair loss, severely deliberating the quality of life of patients. But in the recent years, we have more and more new drugs approved yearly, and the game changer is the use of our immune system. So our immune system have many different cell types that aimed mainly at battling bacteria, viruses, and parasites that enter our bloodstream. But one of those cells, a T cell, also knows how to detect an enemy inside the cell. So it acts like a detector. It can recognize small protein parts on the surface of the cell and target it and kill it. It can detect viruses hiding inside the cells, and it also can detect the abnormal cancer cells. Unfortunately, cancers are smart, and they know how to hide themselves from the immune system. They mutate their DNA rapidly and quickly and mask themselves from the immune system. They also turn off their internal anti-invader response, which makes them invisible for the immune system. What we want to do is we want to shine a big flashlight on the tumor to signal the immune system to come and kill off the cancer. That's exactly what is happening in many of the new, what happens in many of the immune modulatory drugs that we use today, the new approved drugs that we use today. So I just want to say that last year I had the privilege to hear a lecture by Professor Dina Ben Yehuda from Hadassah Medical School, and she treats leukemias, blood cancers, and she said that for many years, up until 5 years ago, she had only one combined chemotherapy to offer to all of her patients. And now she have even more than 7 or 8 options to offer to her patients and tailor it according to her patient's disease. So one of those treatment modalities to flush out the tumor is treating the cancer with viruses. This is a novel approach. And how can we use it? It was discovered as a bystander effect by mistake. Physicians saw that some of their cancer patients that got sick with specific viruses saw their tumors shrunk. And they was wondering what's going on. So what is going on is that the same techniques that the tumors use to evade the immune system actually render them sensitive to the viruses. Viruses now can go inside the tumor cells, infect them, divide, grow, and then lyse them, burst out of the tumor cells without being detected by the immune system and kill them. What is also interesting that it also shines a big flashlight to the immune system to come and kill off the remaining tumor cells that are still there. So this treatment modality is now tried in several clinical trials to do exactly that. But our problem is that the viruses are not specific. They go everywhere, and they mostly go to normal tissues, specifically liver, spleen, and others. And there they are killed by our normal immune system, functioning immune system, and thus reducing the efficiency of the treatment and perhaps also leading to side effects. What we propose, where we wish to connect between the virus, the oncolytic virus that is termed, the virus that we use to treat, and the cancer cells. We want to target them specifically to the cancer cell and create what is called a targeted therapy, or even you can call it a personalized therapy. You know exactly what cancer this patient has, and you use something to connect between the virus and the cancer cells. Several cancer cells also have markers, something that mark them different from the surrounding normal cells. So what we do, we use adapters or connectors, something that connects between the virus and the cancer cells. On one hand, those are small proteins, peptides, that on one hand can interact with the virus, and on the other hand, interact with the specific cancer of that patient. What happens is that this peptide covers the surface of the virus and targets it to the specific cancer cell type. This novel approach was developed in our lab and is now considered a registered patent. We use this technique for CEACAM6 positive carcinoma cells. Some of this, for example, CEACAM6 positive carcinoma cells are present in colorectal carcinoma and pancreatic carcinoma that was mentioned before. So in comparing to colorectal carcinoma that has 5-year life expectancy of 90% if discovered early, the pancreatic carcinoma have only 7 and is usually discovered very late. And you can see in these top images of cultured cancer cells in red that these carcinomas express this marker, CEACAM6 marker. In blue, you can see the cell's DNA. And when we use this specific adapter together with our oncolytic virus, the virus that treats the disease, you can see here in a bar graph, in red, when we use non-specific adapter that cannot bind to these specific cancer cells, the cells continue to grow. The bar graph represents the cancer cells in a culture dish. But in blue, when we use the specific adapter, the cells are killed off in a culture dish, and we can treat this specific cancer. Also in the pancreatic carcinoma, similar effect is seen. Only when we're using a specific adapter that recognizes this cancer cell type, only then, in the blue bar, we can see the reduction in cancer cells. This does not work in non-specific cells. If we use other cells that do not express this marker, we will not see this effect, and cells will continue to grow. So I just want to summarize and to say that we want to use our immune system. This is our best bet to fight the cancer. And we know that cancers hide from the immune system. They know how to mask themselves, and we want to shine a big flashlight and unmask them for the immune system, especially the T cells. But the same system that they use in order to hide from the immune system makes them vulnerable to viruses, specific viruses, oncolytic viruses that can grow inside the tumor cells. We want to use a targeted, directed, specific technique to direct the viruses using an adapter to specific cancer cell types that we choose. This will allow the virus reproduce and grow only inside the cancer of our choice, and then also summon the immune system. I also want to mention that we use this adapter technique also for other treatments, to develop other treatments like in vivo CAR-T and gene therapy for genetic disease like cystic fibrosis. And if you want to talk to us later about that, I will be happy to elaborate. I would like to thank Professor Menachem Rubinstein, here my PI, and also our collaborator, Professor Gideon Schreiber and the Schreiber's Lab, who host me, and also Dr. Merav Maron from Bina and Dr. Elek Chapnik from Yeda, who help us to commercialize this patent and hopefully make it into a real-world drug one day. And in the last 10 seconds, I would like to dedicate this talk to my mom, Ella Gataulin, that died in a car accident last summer. She was supposed to be 60 tomorrow, and She was always supportive about my science, and I hope she's proud of me today. And thank you very much for listening.